seagrass die-offs in Florida Bay, which is a large shallow lagoon that covers almost 3,000 kilometers squared. So we're also interested in the broader implications of our results in Florida Bay to global seagrass decline, particularly with ocean warming and salinization with climate change. So hopefully I can, there we go, okay. Florida Bay can be seen here. It's a region between the southern tip of the Florida Peninsula and the Florida Keys, which is a highly populated series of islands linked by a road. But you can see it's part of the larger Everglades ecosystem, which is the freshwater marsh, the mangroves, and then this large seagrass system, and then coral reefs. The thing about Florida Bay is it's been experiencing these large-scale seagrass mortality or die-off events for actually many years. Upward of 100 kilometers square, particularly the tropical dominant seagrass species, the Lassia testudinum. One occurred in 1987, which is kind of shown here with the hatch marks, and another in 2015. However, there's smaller seagrass die-off events that occur almost annually or every few years. One of the major enduring hypotheses driving these large-scale mortality events is the meristem, which is the growing part of the leaf, the meristem, a leaf meristem, where the leaves are growing. It's a very sensitive area. That is affected by hypoxia, followed by hydrogen sulfide intrusion. It's really interesting. These events occur at night, and the shoots can be seen here as being severed from the leaf meristem. And this denaturing at the meristem causes the live shoots to disconnect, resulting in a large mat of leaves at the surface. And you can sort of see that here. What's interesting is you can see that the leaves were actually alive when this detachment occurred. And so, of course, that makes a big problem because then you get those nutrients recycling into the water column, followed by algal blooms, which then can have what they call secondary die-off events in the system. So fundamentally, as most of you all understand, seagrasses depend on a high rate of photosynthesis and oxygen for their oxygen balance. So oxygen produced by the leaves is released into the water column, but also throughout the plants. At night, however, the plant is very dependent on oxygen from the water column. So the focus of our lab's research the last few years has been linking the ability of seagrasses to internally oxidize their tissues, considering the internal physical structure of the parenchyma and barriers to gas exchange, gas dynamics due to external drivers, water quality conditions, and climate change impacts more recently on stress and mechanisms of resilience. So I've pretty much organize the talk around those areas of research. And so I was going to talk a little about those. The internal structure of a parenchyma is very important as it facilitates the diffusion of oxygen from the water column into the plants at night. And further, this structure also controls hydrogen sulfide intrusion into the plant. So this shows some CT we've been doing, looking at internal air spaces or parenchyma tissue in Thalassia. So this is one of the dominant tropical Atlantic Caribbean species, which is a late successional habitat forming species. But we've been looking at it in a couple other species, tropical species, using micro CT. And you can see this 3D image here of Thalassia leaves that transitions into the meristem. So this is the meristem region here, where during seagrass die-offs, that's where you get the separation of the shoots. And you can see these extensive channels in the vertical rhizome, and they go up to the base of the meristem. Thus, if hydrogen sulfide can get into the rhizome, it has a relatively non-restricted conduit to the meristem. Although the leaf tissue can be less exposed due to this constriction, and you can see this constriction here where you have 33% of the volume of the tissue actually gets reduced at the meristem. But in terms of oxygen, photosynthesis, it's driven from photosynthesis through the leaves, through this whole lacunal system, into the meristems and below ground tissues. So we thought, based on the current literature, that this may be important not only for maintaining aerobic respiration below ground, but to oxidize the surrounding sediment and keep hydrogen sulfide from the plants. So we started looking at whether or not we have oxygen released into the sediment, thinking that that might be affected by salinity or high temperature or other factors. And so we started using these 2D optode sensors to look at oxygen release from the roots. But what was really interesting is we never really found any oxygen released into the roots with these 3D optodes. So this would be this system right here. This is the optode sensor, and these are the roots, and it's under full saturating light for photosynthesis, and you still see no oxygen being released. And you can tell that because these are 2D optodes, so this would be 0 oxygen. So you don't see any oxygen release based on these optodes. So we started questioning that because in the literature, there's a lot of thought that there's a lot of oxygen that's being released from seagrass roots. So we decided we have microsensors. So we started looking at some microsensors, using microsensors to look at oxygen flux from the roots. And we looked at individual roots. And then for some of the smaller species, we looked at bundles. And this graph here shows pretty much what we found there was that as you get closer to the roots, there really was no oxygen, basically confirming what we found with the 2D optodes. And we did a lot of different studies. So we're very confident in these results that we really didn't see any oxygen released right next to the roots. But then once you penetrate into the root, typically the oxygen goes down when you're just in the tissue. So when you would be just in this part of the tissue, the oxygen would actually go down. And then as soon as you hit the parenchyma, it goes up. So we're continuing with this research, and we're examining barriers to oxygen, root oxygen loss. Things like lignin and suberin have recently been found in rice to reduce the diffusion of oxygen into the plants, or oxygen release from the plants, but also sulfide coming in. We're also considering just the distance of the lacunae. So this would be the parenchyma, the airspace tissue here from the epidermis. And it's interesting to think about that because even without any kind of chemical development, such as lignin or suberin, you still have a rapid diffusion of oxygen through the lacunal system. But once you hit these cells here, the diffusion rate of gases reduces like 10,000 times slower diffusion coefficients. So it could be also just the fact that you may not have as much oxygen released just because of this lower oxygen diffusion, and perhaps not sulfide coming in unless it can bypass this interaction with the oxygen going out and the sulfide coming in, that it might not actually get oxidized if you don't have a lot of oxygen coming down through the lacunae. So, so we're really looking at that and continuing with that work. So just in summary, internal seagrass structure is important in allowing diffusion of oxygen through the plants. Zones of constriction associated with the meristem may limit oxygen diffusion at night, and that gas dynamics inside plants appears to be relatively conservative. And that makes sense because you would want to have that oxygen going down all the way to those roots, particularly in a species like the Lassia, where they have those very deep roots. To get that, you wouldn't want to lose oxygen as it's going down into the root tip, so continue to have aerobic respiration and grow. Root oxygen loss may not be a mechanism to oxidize sediment hydrogen sulfide, at least in these 3 tropical species that we've looked at. And then barriers to root oxygen loss may assist in limiting oxygen loss and sulfide intrusion. But due to the complex structure of seagrass meadows, we wanted to examine some of these internal gas dynamics of oxygen, hydrogen sulfide in the field, and how these dynamics corresponded to different environmental conditions. So we use the oxygen microsensors in the leaf, and you can see this is the leaf cross section with the microsensor in it. So we're looking at the parenchyma dynamics in the leaf, and also in the meristem. So in the meristem, we looked at not only oxygen, but we also had a sulfide probe. And so these were done in situ in the fields. So we're looking at the sulfide and the oxygen in both in the meristem, but also in the leaf. And we had a light sensor, and we also had a sonde, so we were able to get water quality data as well. So this it's really difficult to get this data. But when you do get it, it's quite beautiful actually, and it's kind of like taking someone's temperature. So like every time you take someone's temperature, if they're sick, you can see that they have a high temperature. You go to another person, it might be the same. And one of the things that was interesting is sometimes it seems like there's so much variability out there, but when you look at the underlying basic physiology, it's actually very consistent. And we found very consistent results across different shoots and different days. And this is just an example of an oxygen profile over several days in Thalassia testudium in Western Florida Bay, which is a site that has a recurrent die-off. Here you can see the leaves in blue, and meristems are in green, and the water column is in black. And this gray is light, so this is irradiance in micromoles per meter squared per second of tar. And what we can see is a few different things here to note is that the leaf partial pressure of oxygen is about 2 times the atmospheric oxygen saturation. So there's a lot of oxygen that is being produced by the leaves, and a lot of that is then going into the water column. You can see the water column follows the leaf oxygen with a lag. And then you also see that oxygen is getting into the meristem, but it also has a lag, and it also is much lower than what you find in the leaf. And it also does not reach supersaturation, and in this case, where the water column actually did. So these are some really interesting dynamics. You can also see that the oxygen goes very low during the night, which we weren't exactly expecting. But so long as the water column oxygen stayed up, we really didn't see, and there was enough oxygen in the leaf, we really didn't see sulfide intrusion at those sites. We also observed some interesting things about the leaf oxygen maximum. It was able to be sustained until the late afternoon. So this is the light curve of one day. This is the leaf partial pressure of oxygen. You can see how it goes up very quickly. And then it stays at fairly high levels until about 1530, and that was pretty consistent over many plants. This also suggests that when this is then declining from this high levels of accumulating oxygen, it's about 1,000 micromoles. So even though maybe the plants photosynthesize and saturate at 500, it's twice that for the entire plant. So that's a pretty interesting result. So these plants need a lot more oxygen, maybe double the oxygen that you would think they might need for saturation. Also, oxygen goes into the water column until late afternoon. So that's that oxygen that's diffusing from the plant into the water column can be really, really important. Also at nighttime, indicated by the gray bars here, it shows this sort of shoulder of water column oxygen above saturation, indicating, and this is kind of an interesting feedback because you have oxygen from the leaves going out into the water column, but then there's so much oxygen from the leaf to the water column that it then can diffuse back into the leaf at night over a certain amount of time. So that oxygen conditions in the water column during the day is really important in that early part of the night and keeps it from going as anaerobic or as low oxygen, I should say hypoxic, in the night. And so that seems to be kind of important as well. You can also see at night that the meristem becomes hypoxic on several occasions, and we saw that consistently. But but we rarely saw hydrogen sulfide intrusion. And that was kind of interesting because you would think if it's going hypoxic or anoxic, you would think you would have sulfide intrusion. But that kind of brings us back to perhaps the physiology and the oxygen diffusion and the resistance to that diffusion of gases across the epidermis. And so I think more work probably needs to be done on that. I think that there's maybe this dogma that all this oxygen is pouring out of seagrass roots, but I don't really think that maybe that's really what's happening. So one other thing that we thought was really interesting, and also shows the importance of movement of water. And this was the role of the water column on internal leaf oxygen is shown at night with tidal influences. So with incoming tide, it raised the water column oxygen. But what was pretty interesting was we could also see that in the leaf oxygen internally in the leaf. So as that oxygen's coming in from the water column, it's actually also coming into the leaf at night. And then that's facilitating keeping that below ground oxygen levels. So the only time, I know there's a lot on this, the only time we really saw sulfide intrusion was this was in late June, so it's pretty warm in Florida Bay and South Florida where we're working. But this is again the light is in gray, and I want to show you just a couple of things to highlight here. So number one, this blue is water column. And you can see where we had kind of a hypoxic event in the water column. And when that water column levels got really low, the leaf oxygen got really low as well. And then that is when we saw sulfide coming into the meristem. And so you see it was a rapid increase in the sulfide intrusion. But then when you had the light come on and the oxygen in the leaf, you had that being oxidized and fairly quickly. And so there's this importance again of the water column, water quality, but not just in the sense of light, but also in the sense of hypoxia is really important for maintaining the internal oxygen and sulfide, keeping the sulfide out in below ground tissues. This occurred during our July deployment. So I can just show you this when the water column had the highest temperature. So, and it gets pretty warm in Florida Bay. This is 33 degrees C. And I know there's some work out there that's, sort of shows, well, if you have more new paper that came out, like if you have high CO2 with ocean acidification, temperature might not be effect. But I think that temperature is an effect because it really does affect the oxygen solubility. And so when you look at the partial pressure of oxygen, it was very low during that time where we had this highest temperature. And then the oxygen concentration dropped below 1 milligram per liter. So just a couple other series of experiments that we've been running. This was a field experiment also with the microsensors showing, we did some shading experiments, and we showed a series of low light days can also stimulate internal sulfide intrusion into these plants. We also were looking at salinity because this is a semi-enclosed hypersaline lagoon. We do get high salinity occurring. And not only does high temperature and high salinity reduce the solubility of oxygen in the water column itself, but it also affects the photosynthetic rate. So what we found, and this was experimental, we found a lower photosynthetic rate, but also an increase in the respiratory demand. So both of those make sort of a very large imbalance of oxygen physiology for the leaves. And then this hypersalinity, this was a series of light-dark experiments with the microsensors in experimental situation. And there's this sort of relationship between oxygen, photosynthesis at night, oxygen in the meristem, and then how low do you go? So if you have higher salinity, you don't go up as much with photosynthesis. So then you come down even more at night. And then that was when we saw this hydrogen sulfide coming in at this hypersalinity. It's a very high salinity, but that was where it was triggered. And that's where we see some of these major die-off conditions where we had clear water, but hypersaline conditions. So just in summary, seagrasses have a high capacity to raise oxygen in the leaves during the day, 2 times atmospheric saturation of oxygen. Leaves acquire oxygen at night from the water column to internally oxidize hydrogen sulfide. Low water column oxygen compromises nighttime oxygen diffusion into leaves, which can lead to sulfide intrusion into the meristems. Low light increases hydrogen sulfide intrusion into meristems, indicating plant water column feedbacks. And high salinity temperature affects internal tissue oxygen balance, as well as enhances ecosystem oxygen consumption and lowers oxygen solubility. So in order to stimulate climate change and bring some of these results into more management scenarios and a more comprehensive understanding of what we think is happening in seagrasses, and we're applying this in Florida Bay, but also to some other estuaries in South Florida. We've developed a collaboration with Dr. Chris Madden, who works on seagrass management in Florida Bay and has been developing this model. Actually was originally developed in Chesapeake Bay. So it's been around for a while. And we've been using it for defining freshwater flows into Florida Bay from the Everglades in terms of restoration. But it's we want to take this model and integrate more of our understanding and base of the stressors and the physiology into the model, so that it can be more mechanistic and we can understand the water quality and physiological drivers of the seagrass decline and die-offs, and also have some different scenarios for climate change. I'm not going to go into these, but this is just using the model kind of how it is now. If you increase phosphorus like into the water column from, say, repeated hurricanes or more intense hurricanes, which we have here in South Florida, can stimulate the phytoplankton blooms and decreases in light in the canopy, which also can make this above ground biomass decline of the seagrasses. And then we have the hypersaline effects based on our mechanistic research showing how that affects above ground biomass. But what was interesting was increased temperature actually increased photosynthesis and biomass. These are tropical species. But what's missing from the current model is this relationship between hypersalinity, temperature effects, and the oxygen in the water column, and how that oxygen is really important for maintaining or keeping these seagrasses from having these large-scale mortality events. So this is just a spaghetti diagram, but how we're trying to integrate respiration, ecosystem respiration, salinity, hypoxia, and make it such that when we run these models, we are integrating more of the fundamental understanding that we have of some of the mechanisms that we think are really driving the seagrass loss. So with that, I didn't have a chance to really even time my talk, so hopefully it wasn't too long, and I'm sorry about all the delays, but I'll take any questions.
Thank you, Marguerite. Is there any questions? You can also post in the chat if
I have the chat up here.
Could you go back to the last model really quick? It was so interesting, the last slide. And so that's
yes.
that's what you're using to try to integrate like the temperature and salinity effects on the oxygen.
Right. So what we're trying to do is integrate water column oxygen.
Okay.
So This is just one small part of this
Yeah.
very large model that has a lot of different submodels. Like there's a whole submodel
Mhm.
for like salinity, a whole one for temperature and all of that. But what you can see is that those, what we're trying to do is bring in photosynthesis. So
Hmm.
what's the input to the oxygen model would be increases in photosynthesis. The phytoplankton doesn't really seem to have as much effect. It's definitely seems to be driven more by the seagrasses.
Got it. That's really interesting.
Yeah, but then also bringing down the oxygen in the water column due to ecosystem respiration,
Hmm.
temperature effects, salinity effects. And then the other important one, because we're working in pretty shallow areas, is actually the diffusion of oxygen from the atmosphere. As a matter of fact, I
Oh.
was going to show it, but I didn't want to show too much more data. But we had some interesting results of a big storm we had, which is another thing that could be happening with the future with climate change. You get
Yeah.
more fresh water and you get a lot of fresh water all at once. It creates this cap.
Hmm.
So now you go into night, high temperature, high ecosystem respiration, maybe high salinity. And then you have this cap where you don't get oxygen diffusion from the surface, or if you get a very quiescent day where you don't have a lot of oxygen from the atmosphere coming in,
Got
that
it.
seems to have also like reduced the oxygen from what we saw.
That's
Yeah.
so interesting. Thank you.
So yeah, I think there's some interesting cascading effects that need to be thought about.
I think Anitra has some questions.
Yes. What happens after tropical storms or hurricanes in terms of the hydrogen sulfide entering the plant and any mass mortalities or partial mortalities?
So the sulfide's always there. And there's a certain amount of exposure that always seems to be there too, which is interesting. But what I think it is to do with is to do with the oxygen inside the plant. So if the oxygen is inside the plant, if they can photosynthesize, I think they can deal with the sulfide. I don't think, and actually we've done experiments on that too. Like can be pretty tolerant of it, so long as they are photosynthesizing and producing a lot of oxygen and there's oxygen in the water column. So what happens in a hurricane? A hurricane, you get resuspension of the sediments, you get in our case, phosphorus limits, you get nutrients in the water column, which then drives the phytoplankton, and it's the light, the lowering the light that the phytoplankton blooms, I think that has the more of the effect with the hurricane rather than any kind of direct effect of the hurricane on the sulfide itself. That'd be my answer to that.
Thank you.
Brigitte?
Uh, I'm sorry. Hello, Marguerite. Very interesting.
Hi,
This
hi,
is such
Brigitte.
a nice review. My camera is out, but I
Oh,
think I always been teaching the students that the roots maintain a sort of oxygenized zones around them, and that influences the whole microbiome. But from your anatomy results, the roots don't release oxygen, is it?
I really don't think that they do. I mean, there might be some, and I mean, I know that there's a lot of studies that show it, but we haven't been able to find it. Maybe if we had much smaller microcenters, ours are like 100 micron. Maybe if we had 10 micron, we might see a little bit more. But I mean, it's not, at least in these tropical species that are exposed to sulfide. So we are really digging into this a little bit more, and we have looked at some microbial communities, but we haven't done the transcriptomics and all that with the microbes that I guess tell you what they're actually functionally doing. But my guess is that I think that the microbes are keying more into the DOC and and the organics that are released from the seagrasses, and maybe not so much the oxygen itself. But maybe there's a lot of other organisms that are important for oxidizing the bulk sediment, in fauna. Might be more important, right? So if you have a hypoxia event, you wipe out all your infant, then you really are affecting your, your anaerobic and your sulfide conditions. But, I mean, we have felt comfortable enough to actually publish a paper on it when 2020, I guess it's 5. But we are really still trying to find out more about the barriers to oxygen loss. But just the other day, I started thinking about it, and I really think that it has to do with the diffusion coefficients that, they're 10,000 times slower once it hits water. So if you have 4 or 5 cells, so it goes fast through the parenchyma, right? Because gas going through air is just, easy. But once it hits water, right, it's slow, very slow. So even if sulfide was coming in, which we know it is based on the isotopic ratios, we know that it's getting into the roots. We know that it's getting into the rhizomes, not so much the leaves, maybe sometimes in the meristem, but it can be turned into, if there's oxygen there, right, it can be turned into sulfate. It can be turned into elemental sulfur. There are ways it can be denatured. Marion Homer's work shows that. And our isotopic data shows that it's getting into the roots and the below ground, but not so much into the leaves. So there's, I think that the sulfide slowly diffuses in and the oxygen slowly diffuses out across those cells that are not parenchyma before it gets, out into, say, the sediment. And we even looked at in the root rhizosphere, we looked at, okay, well, maybe it's not oxygen in the rhizosphere, which is the little sediment area around the root. Maybe it's not, the oxygen's getting out there, but maybe it's reducing sulfide. But we measured sulfide, we know there was oxygen in that root, but the sulfide levels never went down.
Yeah.
So I don't know, Broderson's paper and a lot of their work and a lot of other people show that there's all this oxygen coming out, but we really haven't seen it. Maybe it's a tropical species thing. Maybe, I don't know.
Yeah, interesting. I will revise my classes. Meanwhile. Thank you.
I think Anitra has another question.
Yeah, one more question,
Sure.
which is, I didn't quite get if you have done this the same process on Serengodium and Halidul and perhaps
Yeah, we did.
Halophila too.
We didn't do Halophila. We did Serengonium. We did Haliduli, and with the microsensors, as well as with the optode sensors, the 2D optode sensors, and we did not see oxygen release from them. When we did try to do some methylene blue, but the problem with methylene blue, it also shows microbes, so it's kind of hard to know if that's real. But when we did that, it was very little bit patchy, and it didn't create these big bulbs like you've seen, like with rice and everything. And there seemed to be a little bit more maybe with Haliduli and Serengonium than Thalassia, but I'm still not convinced. Because if you think about those Haliduli and even the Serengonium, very long, very thin roots, they have to go a long way. And I just I don't know why a plant would want to lose oxygen, if it's growing in an anaerobic environment and it's trying to aerobically respire and grow at a root tip. But
So
think,
was there a difference between the Serengodium and the Halidul?
no, not really. Mm-mm. And then when the microsensor, we did all the same experiments, but my student is writing that up now. I mean, we just really didn't see it. We didn't see it coming out. No, we didn't. And then when we even got into a root of a haliduli, like a big one, and the oxygen, as soon as you get into that root parenchyma, boom, it goes up. So there's a pretty steep gradient there. So you would think that if it was just coming out, that it would be a lot coming out. and we didn't see it. But if it was coming out, then it probably wouldn't be as high in the root, which, yeah, it's an interesting question that kind of needs a little more re-examining at this point, I think.
Thank you.
Yeah.
If there's no more questions, I can ask it one.
Oh, sure.
So thinking about this, because you mentioned turbidity might be like an important factor, and I guess light,
Turbidity?
yeah,
Yeah.
turbidity, and I guess light plays a role in there. So is there any, or have you looked into some latitudinal effect? Thinking about these like very northern latitudes where there's either very long days or very dark days. Is there any effect there? Any difference?
Well, I think there could be. I think it depends on the oxygen demand, like of the system, right? And so at higher temperatures, you're going to get a lot more oxygen demand, and you're going to get higher salinities, right? So maybe in the, northern, and you also in those northern, you also have a lot of tide sometimes, and you can oxidize the sediment probably with just the tide going down and going back up. So I think it's, it's possible if it's very highly organic, maybe if there's a lot of pollution, I could see, yeah, that could be effect. Low light, high pollution, but then you still don't have the high temperature. So maybe the respiration won't be so high and maybe they can survive through the night. I think it's the combination, which is kind of like why we're a little bit concerned about the tropics and some of these semi-enclosed areas that have a lot of organic matter. It's because with climate change, it's going to get more, hot. It's going to get warmer, and you have hypersalinity, and yeah, it's not a good combination. Yeah.
Yeah, but I think it's still like interesting to think about like these tropicalization effects. If later, as you say, with climate change, we will start seeing that also in other latitudes.
Yeah,
But
could be. Yeah. And I wonder if some of these species are changing their morphology. It could be interesting. I think getting back to looking a little bit more about the morphology and like where there's cells and where there's a rhynchoma spatially would be really kind of important, because I think it does have to do with the diffusion of the gases.